The global population has reached 7 billion, accompanied by the growing collective desire for a better and aesthetically pleasing lifestyle. Skincare and cosmetics have become an inseparable part of our daily routine. But are we aware of its dark side? The high demand of plant-based ingredients, driven by the increasing popularity of natural products, is resulting in overexploitation and deforestation. India's fragrance ambassador, the sandalwood tree, is one such example. Indian sandalwood is rich in alpha and beta santalol, making it a highly sought after in global cosmetic and fragrance industries. To gain a deeper understanding of this, we visited the sandalwood oil factory of KSDL in Mysore, where we learned that the factory operates 20 large distillers only to extract a mere 600 ml of oil from 20 kg of tree's hardwood. Their recent challenges include a low supply of the wood itself. This motivated us to contact Marayur and Aryangav Sandalwood Reserve, the largest of its kind located in Kerala, where we got insights of the factor affecting the significant reduction of the wild sandalwood population. This includes natural factors as well as smuggling and overexploitation. We realized that it is important to learn about the high demand for the products derived from the sandalwood tree, which in turn was validated by KSDL factory in Bangalore, a major producer of sandal soap and other products. This strongly calls for an alternative method for production, one that does not strain the environment but is efficient enough to meet the current demand. Global plastic waste generated annually has been a fueling crisis. Only 15% of the 353.33 million tons generated annually are recycled. India tops the list with 9.3 million tons contribution, double that of other populous countries. In our commitment towards sustainability and environmental consciousness, we found the perfect feedstock in PET. Here is where PETal steps in, aiming to provide a sustainable solution to conserve these threatened species while ensuring that the cosmetic industry remains profitable. Utilizing PET as a feedstock, we use engineered Pseudomonas putida to produce santalene, the key precursor, and santalol, the major component of sandalwood oil.
How do we achieve synthesis of sandalwood oil from PET monomers? We choose a suitable chassis and characterize it to grow on TPA and EG as a sole carbon source and introduce required genes sourced from Santalene album. An experimental core is firmly embedded within the design, build, test, learn framework supported by metabolic modeling and structural studies. Now let's navigate the complexities and challenges associated with our goals. To start with, we chose Pseudomonas putida as our working model, as it benefits by having a native terpenoid synthesis pathway and minimal maintenance requirements, making it advantageous. It is known to be a robust biotechnological system due to its ability to adapt various carbon sources. Through our literature review, we found a pre-existing strain of Pseudomonas putida, which was engineered by Dr. Brandenburg to grow solely on PET monomers, terephthalic acid and ethylene glycol. In a meeting with him, he suggested to kickstart the project with Pseudomonas putida TS7EG, which further helped us to complete the project within given time frame. Initially, we faced challenges growing the strain on the desired media as the growth was quite slow, taking over 24 to 36 hours. Hence, we started to optimize the growth on PET monomers by series dilution, supplementing glucose and eventually decreasing its concentration and making the bacteria grow solely on terephthalic acid and ethylene glycol. Now we will understand the sandalwood oil biosynthesis pathway and the genes required for the same: FPPS, SASI, P450 and CPR. The ethylene glycol and terephthalic acid uptaken by Pseudomonas putida follows the pathway as shown here, where it gets converted to precursors of MEP pathway for the terpenoid synthesis. FPPS produces FPP followed by santalene synthase that catalyzes the synthesis of santalene isomers. Oxidation of santalene to santalol isomers is catalyzed by P450 and CPR.
Parallelly, we simulated the entire metabolism of Pseudomonas putida using flux balance analysis. We restricted the most canonical pathway of glucose uptake from our model and introduced ethylene glycol and terephthalic acid uptake and metabolism. This gave us the hypothetical pathway that the bacteria would follow if it were to solely grow on ethylene glycol and terephthalic acid. By this, we created a digital twin of our bacteria, which enabled us to understand and optimize it better.
The first step in the pathway is to introduce the FPPS and SASI gene to synthesize the santalene, a major precursor of sandalwood oil. We obtained and expressed the PCDF duet SASI FPPS plasmid previously expressed in E. coli by Dr. Bolman. The protein expression was confirmed, and the santalene synthesized by Pseudomonas putida showed a characteristic peak at M/Z value of 204 as detected by GCMS analysis. But the abundance in the sample was quite low. As expected, we simultaneously designed appropriate FPPS and SASE bisystonic gene constructs considering the uncertainty with PCDF results. To optimize the expression, we carefully selected the right biobrick architecture such that genes can be incorporated in sets of bisystonic vectors into single bacteria. The linear map depicts a single Arabat promoter, RBS, SASE gene followed by FPPS genes separated by IRES that could be best expressed in our TS7EG strain. The SASE gene can be replaced with any another gene starting with the ATGG under the arabinose promoter using the NCO1 and EcoR1 cut sites, as it's an experimentally verified ORF. One can use the IRES mentioned for a dual expression system and put the second gene using the ND1 cut site. All these features provide modularity to the design part. We designed this part from gene fragments and then ligated into P761 vector. Then we introduced this plasmid into our strain by electroporation and induced with appropriate concentration of arabinose. After induction, we checked the expression of his-tag SASE FPPS proteins by purifying them. Presence of santalene was confirmed by GCMS that was produced by the bacteria, that too at 10-fold increase in abundance as compared to PCDF dual. We were overjoyed at the fact that we could produce santalene, a key component of sandalwood oil, from TS7EG strain growing on PET monomers.
The next crucial step is to oxidize santalene to produce santalol. We identified the enzymes cytochrome P450 monooxygenase and reductase, which form a eukaryotic transmembrane oxidoreductase system and are involved in this final reaction. We realized that the expression of eukaryotic transmembrane protein is challenging in a prokaryotic system. To our surprise, we found that the functional domain of cytosolic P450 CAM, which is native to P. putida, is structurally similar to our santalum album P450. We performed molecular docking of P450 with the same cofactor to identify the catalytic domain and got an assurance that the truncation of the N-terminal transmembrane domain does not affect its catalytic activity. In addition to this, we also showed that the truncation of the transmembrane domain reduces the transmembrane tendencies of our protein, making them more likely to behave as Pseudomonas putida's cytosolic P450 and could be expressed in our chassis. Co-localization of truncated sepia with P450 is necessary for its function. We came across a naturally occurring self-sufficient bifunctional prokaryotic P450 in Bacillus cereus. To our amazement, this bifunctional P450 resembled santalum album P450 and sepia domain when we structurally aligned. A stretch of amino acid sequence, which we termed linker, connected these two domains in a bifunctional P450. Following this architecture, we genomically truncated the N-terminal transmembrane region of both P450 and sepia. Then we fused the C-terminal of P450 with the N-terminal of sepia with the linker sequence obtained from Bacillus cereus and formed a hypothetically self-sufficient bifunctional P450 sepia fusion protein. We successfully assembled our fusion protein insert, but due to time constraints, we could not achieve its expression. Despite the setbacks, we believed that our fusion protein would get expressed, and it would be functional. Thus, we proceeded to explore its multi-substrate specificity. The P450 from santalum album catalyzes the conversion of santalene isomers to santalol isomers in a fixed proportion. We performed molecular docking and developed a hypothetical reasoning for the variation in the abundance of santalol isomers. As shown in the table, the conversion of alpha-santalene to alpha-santalol takes a least amount of energy. Thus, pertaining to our assumption, alpha-santalol is going to be produced more as compared to other isomers. To further optimize our system for higher yield of standard oil, we performed FPA and identified genes DXS and DXR, which, if overexpressed, will increase the metabolic flux of santalol precursors. This result was correlated with Dr. George's findings as well. The uptake of close to 2,282 mg of PET monomers can yield around 400 mg of a mixture of santalol isomers from 1 gram dry weight of the bacteria in 1 hour.
In the rapidly evolving world of synthetic biology, we set out on an ambitious mission to bridge the gap between cutting-edge science and society. Our journey took us across 25 locations, touching the lives of over 6,700 people through numerous meticulously crafted events. We collaborated with We Grow Forest Foundation, a women-founded NGO dedicated to fostering a greener planet, and organized a beach cleanup at Bailey Beach in Trivandrum. We interacted with Kani tribes, educating them on plastic waste management. We expanded our reach to the Kudumbashree Women's Group, discussing women in STEM and sustainable menstrual products. Through multilingual pamphlets, we raised awareness about plastic pollution and our project's goal. We celebrated World Environment Day with young children by planting saplings and conducting a coloring competition. We unraveled the mysteries of DNA and educated children on tackling the plastic waste crisis. Our standout accomplishments, the Synbioloteria game and a captivating synthetic biology book, transformed complex concepts into an engaging experience for young learners. We organized the BioBricks Innovation Competition to nurture critical thinking. We also engaged college students through game nights, quizzes, and trashing. We organized a project reveal for science enthusiasts at our institute. We collaborated with the Biology Club to host Rhetor 5.0, our annual symposium that brings together experts in the field of biology. The Synbio Conclave united experts from across the globe to share innovative research and explore solutions to pressing global challenges. Durga Shakti, an iGEM alum and a drag artist, gave a talk on their path in science, drag, and art. We visited sandalwood oil factories and sandalwood reserve forests, distributing project pamphlets and gathering valuable insights. Our team also engaged with local plastic waste management facilities to understand plastic management better. Our online reach through photography contests, documentaries that told compelling stories, and social media posts captured wider attention. The All India iGEM Meet allowed us to connect with iGEM teams from across the country, exchanging ideas and exploring collaborations. Through Biogenesis, we worked with IIT Bombay and IISER Kolkata iGEM teams to organize a symposium focused on biotech startups. In our Send Trails and postcards on sustainability, we collaborated with iGEM teams across the world. Along the way, we have witnessed students light up with new understanding, seen communities engage with science in ways we hadn't imagined, and felt the excitement of being part of something bigger. Our commitment to bridging the gap between science and society has only grown stronger, and thus it has been an invaluable learning experience for us as well.
To assess the real-world implementation of our project, we focused on two primary issues: the depletion of sandalwood oil and rising plastic pollution. We analyzed the life cycle of essential oils and compared traditional production methods with various upcycling techniques for plastic. This comprehensive analysis reinforced our problem statements. Engaging with experts like Dr. Johanna Sadler and Dr. G. Suresh Kumar was instrumental in shaping our upscaling strategy for PETal. Following Dr. Sadler's advice, we decided to create a modular working model to optimize our system for industrial conditions. Later, incorporating the suggestions of Dr. Suresh Kumar, we ideated the development of an airlift type bioreactor suitable for producing santalol. Finally, we decided to develop a synthesis unit which could process PET plastic waste and synthesize santalol. Initially, we will segregate and sterilize the plastic, subject it to monomerization, and supply the resulting monomers to the bioreactor for santalol production, followed by extraction and quality assessment. To analyze marketability as a B2B company, we identified our market niche through stakeholder interactions with government organizations, startup incubators, and cosmetic companies. This was complemented by a detailed market analysis and consumer surveys. We established developmental plans using SWOT and PESTEL analysis, outlining de-risking strategies and a clear action plan for patenting our technology. The cost estimation of PETal sandalwood oil revealed a significant reduction in the price when compared to the current prices of sandalwood oil in the market. Later, we outlined a 10-year financial plan aligning with PETal's future timeline, assessing both the direct and the indirect impacts of our project. PETal is not just a cosmetic product. It's a shift in the perception of waste plastic as a feedstock for bacteria in biotransformation processes. As an undergraduate team, we hope that our project inspires students and researchers to pursue similar research endeavors and realize that anything is possible. Towards the end, we would like to thank our director, Professor J. Murthy, our PIs, Dr. Vijay, Dr. Ravi, the School of Biology, IISER Thiruvananthapuram, and the iGEM community for their unwavering support and help us lay the foundation of PETal. We owe our success to everyone who helped us along the way. This is PETal signing off.